episode 286 of the Stem Cell Podcast, Neural Lineage Identity, with Dr. Marius Wernig. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying this podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Not going to do much better than the one we have today, though, who is Marius Wernig from Stanford University. He's on the podcast to talk about his research on the molecular determinants of neural cell fate decisions. We're going to get around to that. But first, we got our usual roundup of recent highlights in stem cell news. That's coming right up.
Yes, but first, and this is important news, big news, one of our favorite news pieces of the year, of course, is to advertise the iPSCs annual meeting, which is coming up June 11th to 14th in Hong Kong. The deadline for abstract submission has been extended to January 28th, so submit your abstract now at www.iPSCR2025.org/abstracts. And yes, we cannot wait for iPSCR across the world in Hong Kong, hoping to see our listeners who listen to the show based in Asia, and can't wait to see you all in that beautiful part of the world. So starting things off, this is more of a news piece than anything else. I mean, it's based on something we've talked about already here on the show, which is transplantation of a genetically modified pig heart into a live human. I think we covered this as a news piece like a few months ago, and this is certainly something that caught the scientific community and the broader community by storm. You might remember that there've been a couple examples now of folks who have received these modified immunocompatible pig hearts for transplantation in a situation where there was no human heart available. So the immediate goal is that this serves as a bridge organ to a true human heart, but the long-term goal is that these organs, these immunocompatible pig hearts, could be long-term solutions to the organ shortage crisis. Of course, the heart is just one example. You can think about applying this immunocompatible organ technology to whatever organ you're interested in, except for the brain, of course. Yeah, I'm pretty sure you don't want a pig brain. Maybe you do, Dalen. I'm pretty sure I don't.
I can do worse.
Yeah, but this is we're talking about the heart here. So this is the paper in Nature Medicine that was basically recapping the finer scientific details of that second transplant. This is pioneering, amazing work led by folks at the University of Maryland School of Medicine. In particular, last author here is Mohammed Moeddin. First author, Bartley Griffith, but this is just, as you know, with anything heart transplant related, this is just a massive effort with 30 plus people in on this paper, and I'm sure a lot more people involved with the project. So here they wanted to improve on the results of that first patient that received the immunocompatible pig heart. They selected a healthier recipient and actually did a more sensitive donor screening for potential zoonotic pathogens. So this is always a concern when you're transplanting a non-human organ into a human, right? Are there any resident pathogens from that non-human organ that could be reactivated in the human setting, right? There are these porcine endogenous retroviruses, for example, other zoonotic pathogens that might be in these organs. So they did a extensive screening for that, and then they transplanted a 10 gene edited pig heart into a 58-year-old man with a progressive, debilitating, inotrope dependent heart failure due to ischemic cardiomyopathy. And again, importantly, he was not a candidate for standard advanced heart failure therapies like, heart transplant, all this kind of stuff. So this is really a last case scenario. And he was ultimately maintained on a co-stimulation blockade-based regimen to modulate the immune system. That's the biggest issue with these cross-species transplantations, of course, is you want to avoid that immune storm that inevitably would happen if you transplant a non-human organ into a human, right? So they mentioned they did an extensive immune regimen, anti-CD40L, tegopubart, a bunch of drugs, the whole gamish, the whole cocktail of anti-immune compounds to really suppress the human immune system. Initially, the xenograft functioned pretty well, and we talked about this, for a few weeks. The guy was doing all right. Really excellent systolic, diastolic function in the heart during the first several weeks post transplantation. I remember, I think he was actually able to walk around, ambulate to some capacity, which is pretty incredible. But and again, this is the major problem with this particular field. The xenograft, the pig heart, ultimately developed a rapidly progressing diastolic heart failure, biventricular wall thickening, and complete loss of systolic function because of these basically immune rejection, right? And this necessitated the initiation of, basically ECMO, extracorporeal membrane oxygenation, just to keep this guy alive on day 31, 1 month after the initial transplantation. So it was doing all right. The heart was doing great for about 2 weeks, and then some of these immune problems set in. Um, you know, it, it's it, it's a great advancement here. There were some setbacks, of course. The patient ultimately chose to transition to comfort care after 40 days. Histology actually didn't reveal substantial immune cell infiltration, but also suggested that there's some capillary endothelial injury with some edema, fibrosis. Um, Importantly, there was no evidence of porcine megalovirus replication, those, pig viruses that could get reactivated. There was no evidence of that in the xenograft. So the real goal for the next iteration of these transplants is to overcome this obstacle of antibody mediated rejection. All right, that's what they assume was happening here. But regardless, I mean, this is a tremendous advancement for the field, for the heart transplant, organ transplant field. This was just such an honorable and commendable thing that this individual did, and this care team, team of doctors, did presented here in advancing organ transplantation. Just such a tremendous sacrifice, of course, but something that's going to progress this field. And the dream, again, ultimately, is that these sort of transplants, xenotransplants, become widespread within the next 10 years. And at the rate this field is progressing, I don't think that's out of the realm of possibility.
Yeah, this was an amazing advance when we talked about it initially. It's an amazing advance because, like, even an incremental advance in this heady field is really world changing. But I'll be honest, this was a bit of a gut punch for me because, the intent, I thought the design was great. We got so much information, and that's why that pushed the field forward. Not to mention, as you alluded to there, like what goes for the heart is going to go for all these other organs, right? Because if anything is going to be, notwithstanding the brain, as you joked, but maybe some other organs for some other reasons. But I would think that, like, the biomechanical element of the heart, and complexity cell wise, and the vascular perfusion, all that, not to mention functional, how much the heart works. I would say that this is really the anything that you can accomplish here will be applicable across many organs. But as I said, it was a gut punch for me because the intent here was carefully designed to address, I thought, what the authors acknowledged was a little bit less fleshed out in the earlier study, which was like that xeno element, the zoonotic viral element. And that was like one, the major, there are two major intents as far as I see it from the genetic modification. One was the zoonotic, and one was the immune rejection, right? All the gals, right? So if the idea was to address those, I would say it looked good in terms of zoonotic viral transmission. But while I had taken for granted that immune rejection and there was no cellular infiltrate here, which I think is key, that antibody-based pathology, I thought was really, that bummed me out. I thought it's really disturbing. because, the capillaries, like really at like the most rudimentary level, where every cell is like 2, 3 cells away from a capillary. And if that integration is, and in a very specific way by antibodies, not cells, is being undermined, then that it's kind of like, oh wait. And I didn't read it as closely as you did, and I'm sure the authors and a lot of other thinkers have kind of used this as fodder, be like, okay, here's the next obstacle to surmount. But from my reading, it was a little bit murkier. It's like, wait, how is that? What's the problem here? I couldn't put my finger on why the capillaries were having the antibody deposition, and if that was the root of the pathology. And I don't think that anybody really could definitively, but it was a little bit unsettling to me because just when I thought like it was all cinched up from the immune standpoint, it's like, oh wait, here's a problem, an unknown unknown, a problem I didn't even know we were having.
Yeah, I'm with you. I think this is going to be a big hurdle to overcome because, the lack of the immune cell infiltration is great, but the antibody issue, I think, is going to take a lot of work. Maybe, they're already working on this in some capacity, and maybe it's just a matter of some additional genetic modifications. I mean, this is a pig heart with only 10 different gene edits here, but to modify that antibody activity is going to be pretty tricky. Regardless, this is an again, doing something like this and getting this individual to survive for even 31 days, a whole month, is something that was just not feasible, not possible a decade ago. And it's thanks to advances in genome editing, especially with CRISPR genome editing, that enable these pig hearts, these modified pig hearts, to become a reality. And with continued advances and rapid, effective genome editing, maybe it's going to be easier to make some of these more complex modifications of the immune system. Focusing more on antibody cascades and so on. So very excited to see where this field goes. I remember when I was at Duke, one of the first essays I ever wrote in my, I think, research ethics class or something as a freshman was on this topic of xenotransplantation. It's something that's excited me for more than a decade now, and it's going from science fiction to science reality very quickly.
Yes, and I don't want to be too dark about it. I think, as you're saying there, the biggest breakthrough has been made, and now it's a matter of tweaking, as you're saying, with the we found a new problem, we'll tweak it. And when you're doing 10 modifications, what's 15? What's 20? When we're talking about like a breedable, expandable, you got infinity organs effectively. And as you said, we're talking about even right now a bridge organ, right? We don't need necessarily to make this heart last forever. 30 days already isn't a major breakthrough. But moving on to very, very near neighbor, the lung, right? And I think what this story tells you is how complex the integration of this case, in this case, a whole organ is. But also, I think it underscores the complexity of all those constituent cell types that make up the functional components of the organ, a lot of that being capillary there, endothelium. In this story, we're talking about lung endothelium. I love lung endothelium, but in the lung, the endothelium is a major business end of it, because that, combined with the epithelium lining the alveoli, that's like the medium of gas exchange. You go from air into the bloodstream via that epithelial endothelial interface, right? And those two cell types share this fused basement membrane, and are highly susceptible to injury, as you might expect. They're right there, exposed to the air. So in order to repair lung injury, you need to reestablish those cellular components in order to reestablish gas exchange. And while it's expected that regenerative processes, like that process, like most processes, shares some characteristics with like developmental, in this case, alveologenesis. Not many group studies have directly compared developmental and pathological regenerative alveologenesis, and particularly in like a longitudinal, long-term way in the context of injury, to see like what's the order of operations as those fundamental functional cell types of the lung, how they recover from injury. So to fill that gap, this is a story in cell stem cell from the lab of Edward Morrissey, who's at UPenn. And what they did is, we talk about a lot of atlases. The reason I chose this is because, as we've talked about recently on the show, we're getting now into these more specialized atlases, right? Or of a whole organism, for example, a zebrafish, and a lot now we're doing these atlases of like pathology, and in this case, like a longitudinal atlas of single cell transcriptomic atlas, combined with a lot of lineage tracing of the progenitor cells to look at lung regeneration specifically after influenza injury. That's the modus of lung injury they chose, but I think it has really strong implications for just general lung injury and lung disease, and I'll come back around to that. But what they found effectively, and there's a lot of details here in terms of the constituent cell types, but the bombshell takeaway is not just that there's like this whole catalog of cell types. There's these the AT1, 2, and the endothelial cells as they recover and differentiate from progenitor to more functional cells. There's all these transient, like intermediate states, but they persist. In this case, in the mouse for like a year, which is effectively forever. And that was a bombshell takeaway here. Is that, wow, lung injury is not like something you bounce back from, like maybe you would think liver injury, where the liver is indistinguishable 4 weeks after like resection of a big chunk, right? In this case, at the cell level, at the molecular transcriptomic level, there's these persistent changes that just hang around, indolent. In particular, these injury-induced capillary endothelial cells called ICAPs, that arise after the injury and just persist. They just hang around. And they're this strange hybrid with similarities to both developing endothelium as well as diseased lung endothelium. In particular, they have this inflammatory signature that's seen in patients that have chronic obstructive pulmonary disease, right? So the conclusions of the authors here that this may be a model on this persistent lung injury that's recapitulating aspects of emphysema, right? And specifically in a very chilling way, causing these transcriptional intermediate weird cells, injury-induced capillary endothelium that hang around forever. And it really makes you think. I mean, particularly in the news at the time of recording, David Lynch just died of emphysema. COPD affects millions of people. And the etiology is not really well understood. And here is a bit of insight into a little bit of scary insight into how that disease may have its roots really early in lung injury that could happen distally even years before.
Yeah, I think a lot to unpack here. This is one of those longitudinal atlas studies that have been all the rage in stem cell biology and developmental biology and disease biology or whatever. It's more than just doing the atlas and doing the single cell. It's what you learn, what you glean from these large data sets. And I'm going to talk about a paper shortly that's along those same veins. When you were describing this, uh, it's funny that the topic of COVID didn't come up here. And if it was 2 or 3 years ago,
Hmm.
the insult here would have been SARS-CoV-2. So I guess it's an interesting reflection on how the field has, I don't want to say moved on, but maybe even gone back slightly towards influenza, because influenza hasn't gone anywhere, right? Regardless, this idea of transient states, plastic states, comes up again and again on the show, and this is just another example of that. How injury might induce some of those transient states in this situation, in the endothelial cells. And you're right. I'm curious about how broadly applicable an observation like this might be to other forms of lung injury, COPD, lung cancer, SARS-CoV-2, and COVID-19 infection, you name it.
Yeah. I mean, it remains to be seen. This is all in the mouse, of course, and while the transcriptomic hallmarks may be similar, it remains to be seen if this is something that's happening in humans or in normal or disease states. But you mentioned COVID, and I'm glad you did, because that was the thing underlying COVID the whole time, right? Yeah, it was a disease of the lung, but it was a vascular disease. When you saw all these complications in other parts of the body, a lot of the convergence of thought, at least early stages, and who knows, there's been many iterations of COVID that may have different tropisms, but the vascular tropism and the pathology centering around the vasculature with the COVID, I thought was specific. And then you see a study like this, which shows that injury causes endothelium to go berserk and just chill out forever, like a bad senescent cell. Pretty chilling, Arun. And the one bit of good news here is that, hey, maybe it doesn't apply for human. But more seriously, is that understanding one, how these cells come about is one pathway, but also knowing that they exist, like senescent cells. Maybe they can be targeted, maybe getting rid of these, bad actors or weird cells, as I'm calling them, injury-induced capillary endothelial cells. Maybe that, like the whole anti-senolytics, might be a pathway to treatment for these, degenerative lung diseases.
Yeah, man. Well, I guess easier said than done. In this situation, you're talking about targeting endothelium
Yes.
specifically and selectively eliminating that.
Yes.
But
We're not going to do it for me. We're going to try that on some mice first.
yeah, we'll try it on the mice first. Moving on to another story. This one's in Nature, but also another Atlas story, a single cell transcriptomic profiling story. This is looking at not so much disease. It does have a disease element to it, but it's looking more at development. And we're shifting from the lung to the brain here. The title of this paper is Molecular and Cellular Dynamics of the Developing Human Neocortex. This is coming from Arnold Kriegstein's lab, who's just an incredible pioneer in brain biology, developmental cortical biology from UCSF. First author here is Li Wang. We have learned so much about the development of the human neocortex, the human brain, in large part because of studies from groups like the Kriegstein Lab. It's a dynamic process. The development of the neocortex has a bunch of complex cellular trajectories, interaction. Brain development is just it's still a black box to this day, even though we're starting to crack open that black box. And to crack open that box a little bit, they did a ton of single nucleus chromatin accessibility and transcriptomic analyses from 38 different human neocortical samples, which encompass both the prefrontal cortex and the visual cortex. So that's kind of cool here, just different parts of the brain, some important parts for sight, for example. Importantly, these samples are spanning a bunch of different developmental stages in the brain, from way early first trimester samples to all the way to adolescence. So quite a range here. In parallel, they've performed spatial transcriptomic analyses on actually a subset of the samples to show where those cells are moving to, their overall locations and dynamics in that way, and spatial organizations, and figuring out how they're communicating with each other. So this is a broad data set looking at intracellular communication, spatial organization, and from that, they cataloged cell type specific, age specific, and area specific, because thanks to the spatial gene regulatory networks that actually underlie neural development and neural differentiation. So they did all the single cell omics here to figure out how different cellular states and dynamics work in the developing brain. But like we talked about again and again, it's more than just doing the seek, it's about applying it and making new conclusions and developing new hypotheses, right? So they combine the single cell profiling, progender purification, lineage tracing experiments to untangle different relationships among the progender subtypes that are actually causing neurogenesis and gliogenesis. Specifically, they identified a tripotent intermediate progender subtype. We're talking about transient states, right? In your previous study. And here we're talking about transient states in the developing brain, this unique progender subtype that they called tripotential intermediate progender cells, or tri-IPCs. And they think these cells are actually responsible. They're extremely important because they're responsible for local production of GABAergic neurons, oligodendrocyte precursor cells, and astrocytes. Wow, that's an incredibly powerful cell type there. And, for me, it's like, how could we recapitulate that in a dish? I mean, I'm just an in vitro biologist, right? But is there any way you can take that population and replicate it in a dish? That would be extremely powerful. And then this is the disease modeling element that I was alluding to in the beginning there. They actually did a profiling and a comparison to glioblastoma cells, which resemble these tri-iPSCs at a transcriptomic level. So this is, I think, very intriguing. It's suggesting that these cancer cells, glioblastoma, of course, a terrible cancer of the brain, but these glioblastoma cells might actually hijack some developmental processes to enhance their growth and heterogeneity. It's exactly what we were talking about in your story, right? These cells in the disease states are reverting back to more of a progenitor state to perhaps improve their proliferation or regeneration or whatever. There's like a reversion that happens in these settings of disease and injury. So they also one of the last things they did here in this huge data set was combining their atlas data with large scale GWAS data, genome wide association study data, to create what they term a disease risk map, highlighting different regions of the genome that might be associated with enhanced propensity to cause autism spectrum disorder in second trimester neurons. So just a massive, massive data set here, looking at brain development, cortex development across years of development, not just in the prenatal stage, not in the embryonic stage, but way on into adolescence. They teased apart this really cool tripotential intermediate progenitor cells, which gives rise to multiple really important populations in the brain, and then made some astounding connections to how some of these diseases of the brain, like glioblastoma, might have similar phenotypes and similar hallmarks to some of these progenitor states as well. I think there's a lot to unpack here, but the good news is, anybody can access this data, and that's the beauty of these atlas studies, right?
Yeah, this is Atlas on steroids. More in the applications, as you're mentioning there, because, starts with the Atlas, right? 38 human samples ranging from first trimester to adolescence. That's crazy. And then, the developmental biology, and then the cancer relevance, and then the neurodevelopmental disease relevance. Like, yeah, as you said, this is everything. Massive study, probably uniquely capable, the Creasing Lab, of pulling this off. But I want to come back to that developmental and the spatial, because, we talk about, another atlas, right? And this has all the other elements, which you couldn't even make that claim if it was just another atlas. But here, I think it illustrated to me how you really can't get enough of these things. I'm working in trying to make a new cell type now in the lab. And you look for the embryologic correlate, right? Or you look for the fetal correlate, and there's never going to be the data set that converges right on your cell, because it's in vitro. So the more the better. And here, extending that into the spatial element, I think we're going to see now all these atlases coming out, obviously, incorporating the spatial element, because the niche is so important. We have such a monolithic thinking, or not we, I used to have such a monolithic thinking about differentiation, that you take cells along a path, along a line. But the truth is, you got to create the whole neighborhood, right? You got to create the niche oftentimes in order to get the cell that you're targeting. So understanding these cells in context with the spatial, I think that I'm not a brain guy, but as a developmental biologist and a guy who's focused on the diff, that's where I'd be looking. I'd be looking at these neighborhoods, not just the cells, but their neighbors. And I think that's going to be the value of this to a unique cohort that's not so disease focused, just trying to understand, how do these cells come about.
Yeah, absolutely. And we've talked about it a lot in the show of the importance of the niche. I mean, you're not a brain guy, but you're a blood guy, and you can appreciate the importance of the the niche in specifying blood cells and hematopoietic stem cells and their progeny. So you're right. If we want to replicate those mechanisms in our diffs in vitro, we really have to figure out, using spatial, for example, how these cells interact with each other in like a 4D context, not just looking at transcriptomic data, but also just their location relative to each other, and the fourth D, which is time, right? How these interactions change across all the way from first trimester all the way to adolescence. And I think you're right. It takes a very it's sad to say, it takes a high profile lab to do a study like this, because they're the ones who have the resources, the computational power to do something like this. And of course, the samples are the key in all of this, getting access to those early developmental samples, human samples of the early developing cortex. That's really the key to all of this, right?
Absolutely. And here at the apotheosis of the field here, talking about, fetal to adolescence, brain development, neurodevelopmental disease, cancer, everything. The last story of the Roundup is taking it back. But along the same line, right, of what are the factors, what are the forces, what's the niche, what drives these cells into becoming what they're going to be and specializing? And at some level, all of us walking around, we're just a product of a bunch of nuclear and chromatin rearrangements. And I joke, but in a very real sense, that's what it is. It's the push and pull between the order of development and the entropy, and that's what defines that developmental stages, the progression across those stages, order versus entropy, and time. And it's illustrated in many different milestones of the earliest stages of development, from the earliest maternal zygotic transition, and then of course the progression from a totipotent cell to a primed pluripotent cell by way of that naive precursor, right? And this is something that's it's very real to us as stem cell biologists, not just for the developmental insight, but in vitro, when we culture our human pluripotent stem cells, they exist on this same continuum from naive to primed. And you can take primed cells and make them into naive, right? By putting them in specific culture media. And we understand that from like a practical methodological standpoint, but the mechanisms underlying that that allow that conversion from prime to naive, that's not really well understood in cell culture nor in development. The changes happen really rapidly, and as I said, not really well fleshed out. But there are models. There's the idea is that you get this relaxed, less repressed embryonic chromatin, right, which has more accessibility. It's been shown by like chromatin accessibility assays. Also, 3D structures of chromatin have shown that indeed at these earlier stages, it's like more open, physically open chromatin. And that tracks with the whole idea of development, right? The zygote begins as this epigenetically very simple, open, flexible chromatin. And then as development progresses forward and cellular diversification, specialization progresses, you get this more restricted and compacted 3D structure of the chromatin as all these things home in and attach. So one thing, and we get that, right? I think everyone has come around, and epigenetics is probably like the discovery of, I would say, the last 25 years, epigenetics and the role in gene regulation from a molecular biology perspective. But we get that, but we don't really pay a lot of attention to the nuclear matrix scaffold, okay? I mean, I don't even know what that is before I read this paper. You talk about the nuclear matrix scaffold, you know what it is? It's all those RNPs. When you're looking at your RNA-seq data and there's literally hundreds of genes that you just skip, it's that. That's the nuclear matrix scaffold, a complex mixture of protein and RNA. And based on the hypothesis that that structure is really a factor in governing pluripotency, and based on the hypothesis that the increased complexity of that nuclear matrix scaffold may be governing the transition, the labs of Dongwei Li, Yiming Li, and Andrew Hutchins in China, they set out on this hypothesis that the nuclear matrix and scaffold may be like fundamentally necessary and or sufficient to dictate those fates. And they focused on one particular ribonuclear protein that they found was not expressed in or lowly expressed in pre-implantation embryos, but higher in mouse and human primed PSCs for a candidate for that naive specific factor. And they found this HNRNPU, a lot of letters. It's a factor that binds both DNA and RNA to anchor chromatin-associated RNAs to DNA. And that controls a range of functions, including splicing, chromatin organization. And what they showed here is that when you target that, when you disrupt that HNRNPU, or a target of a partner of that they isolated by co-immunoprecipitation and mass spec, they found this partner matrin-3. They show that if you disable either of those, you get primed HSCs adopting features of naive pluripotent state, kind of reverting a little bit, showing that it leads to increased chromatin assembly, reduced DNA contacts, increased nuclear size. At the mechanistic level, they show that there's these cofactors that anchor promoters in prime-specific genes. They anchor those promoters to polymerase 2 to promote their expression. So it's a lot of like basic biology. It's a nature cell biology story that ostensibly is about pluripotency. But for me, I love this idea. I love any idea where you take something that no one was even looking at and really on a hypothesis-driven study here. It's not like they did a big screen and this thing jumped out at them. They went out there and said, you know what? No one's looking at the matrix. Let's talk about the matrix. And now that they show, it seems kind of obvious, right? That like the structure, the ties that bind the factors to the chromatin, like, yeah, of course, those will probably have some role in governing gene expression. And I think, I mean, as you might expect, I'm going to say this opens a door to everything we think about in differentiation event. Now I got to look at all those RNPs. I mean, no thanks, but somebody ought to, Arun.
Yeah, it's not going to be me either. Don't worry. Yeah, this is just epigenetics and really epigenomics unveiling a new aspect of pluripotent stem cell maintenance and in particular maintenance of the naive state. It's an exciting study. It's kind of scary in a way because I think a lot of us traditionally trained cell biologists, stem cell biologists, we still are perhaps a little I don't want to say scared, but naive of the power of epigenomics and the non-traditional DNA part of the cell, right? And regulating cell behavior, differentiation, reprogramming. We say, I think it's almost like a crutch in a way. If we can't explain something in terms of how the cells behave, we can, stock it up to epigenomics. Oh yeah, it's probably chromatin dynamics or epigenomics or methylation or something like that. But it's critically important. And I like how they narrowed it down to a particular factor and it's HNR and PU. And if regulating this particular transcriptional cofactor is easy enough, and it seems like it's relatively straightforward, then perhaps this brings this dream of accessible naive state biology to more of us, right? I think a lot of us are trying to dive more into the naive side of things in terms of naive pluripotent stem cells, and if it's just a matter of modulating this factor, then maybe I will jump into the deep end. I don't know.
Well, I, like you, was cowed by this study in just the way you're saying. We think we have the system under control, and they're the RNPs are laughing at us. But it also, I think, underscores how amazingly powerful some of these methods must be if they can override all of this, fabric, all the fundamental biology and infrastructure that's in the cell and set up to support one fate and not the other. To override that with a handful of transcription factors really speaks to the power of those factors. And I know someone who could speak to the power of those factors too is our next guest. Speaking of reprogramming, someone who has a unique insight, maybe not into all those RNPs, but into the potency of exogenous transcription factors and driving cell fate, even in committed somatic cells. And we know who that is. But before we get there, I got a quick message from STEMCELL Technologies. STEMCELL Technologies invites you to find a pathway into the peripheral nervous system with STEMDiff Neural Crest Differentiation Kit. Whether you need Schwann cells, sensory neurons, or sympathetic neurons, STEMDiff Neural Crest Differentiation Kit enables high purity generation of neural crest precursors from human pluripotent stem cells. You can learn more at www.stemcell.com/NCKIT. All right, you guys, as mentioned in the open, we have a special guest on today's episode, a real legend, I think it's fair to say, in the field and a leader. Dr. Marius Wernig, who is professor of stem cell biology in the Department of Pathology, also primary member of the Institute for Stem Cell Biology and Regenerative Medicine at Stanford University. The Wernig Lab is interested in pluripotent stem cell biology and the molecular determinants of neural cell fate decisions. They were the first to generate functional neuronal cells reprogrammed directly from skin fibroblasts, which he termed induced neuronal cells. The lab is now working to develop novel therapeutic gene targeting and cell transplantation-based strategies for a variety of monogenic diseases. Really, he needs no introduction, but thank you so much, Marius, for joining us on the show today.
My pleasure. Thanks for having me here.
Yeah, welcome to the show. Thanks for being here, Marius. It's been a pleasure getting to know you and your lab's work over the years. I mean, your lab is, like what Dylan said, interested in the mechanisms that define neural lineage identity, focusing on transcription factors, chromatin biology, and it's been a personal pleasure as well as a former fellow member of the Stanford Stem Cell Institute. So thank you for your mentorship in advance. Before we actually dive deeper into the direct reprogramming side of things, why don't you actually just give our listeners an update on what your lab is working on right now, I guess, in your own words.
Oh, yeah, right now. Yeah, we continue to be fascinated by transcription factor programming and to really exploit this really, what we think, powerful combinations of transcription factors to program cells into specific subtypes of neuronal subtypes, but we are expanding also on glial cells. So brain cells all together is really remains our interest. And that comes along with, of course, trying to still understand, how this actually works. We on the one hand, we are engineers to try to engineer new cell types, or trying to make existing cell types from stem cells. But at the same time, we're also really fascinated by the process and want to understand, like biologists, so to say, or investigative scientists, to understand how these transcription factors interact with the chromatin and how they actually induce the specific cell types that we then see. And another aspect of the lab is that to then use these cells, and we want to use them for two major applications. One is to develop, really high quality human cell models to understand neurological diseases. And most recently, we actually have embarked particularly on neurodegenerative diseases. And the other big aspect of how we could hopefully use the cells in the future is by actually developing cell-based therapies for transplantation type approaches.
Yeah, I mean, that was the original idea, right? And why it was such a bombshell as like an advance, right? A material advance, like the technical advance was like, all right, yeah, shortcut, forget the diff, maybe even in situ reprogramming. All these kind of possibilities emerged from that seminal finding. But like, as you're describing there was so much biology there too, which you spent, more than a decade unpacking, and now circling back to this original application, which, I mean, we've been going after all along. How do you use these cells to advance not just our understanding of the biology, but also to tackle disease with many different approaches? And for me, I mean, this is a big deal because I just can't even believe that I haven't met you before this day. I've been in your orbit for like about a quarter century now. And looking at your publications in preparation of this interview, I didn't know before that, but I was reminded of what a OG you really are, so to speak. I saw this publication in Nature Biotech with Jamie Thompson, like, the godfather back in 2001. I mean, that's early days, just a few
A while
years,
ago.
very long while. But what's even more impressive is that was right after like the cells really came on the scene, right? 98. And you were already on track then, developing protocols for differentiation of human ES-derived NPCs in that case into neonatal mouse brain. I mean, that was pretty advanced stuff at that stage. You were getting the cells and putting them in a, chimeric model in the brain. I mean, this is way before Goldman. So big deal.
This was the first paper to show neural differentiation of human ES cells.
Yes, the first. I mean, you've got a lot of those firsts in your cap there, because you've never really shied away from these ambitious targets, and you've hit square on the bullseye on a few of them. Probably attempted many more. I'm sure you have a long list, but the field really has progressed. Talking about it. 25 years, as you said, there's been so much progress. For you, there's a million things I'm sure you could talk about, but what's one thing that you've been most surprised by, or impressed by, or was most unexpected when you set out?
Well, it's really the reprogramming. I mean, the first time I saw iPS cells when I was a postdoc in front of in the dish in front of me, looking through the microscope, I probably couldn't believe it.
There were a lot of skeptics.
Yeah, I mean that was, and then of course our sort of the main finding of the first main finding in our lab when I started here at Stanford, when we saw the first neurons. Maybe not quite as surprised because we had seen the iPS reprogram before, and was really like our working model, so it really should work. So we were sort of expecting it. But those two examples. So the first time I saw the iPS cells in the dish, and they just looked like ES cells. I couldn't believe it. They were exactly behaving like ES cells. We had some stuff going, in Rudolf Lineage's lab where I did my postdoc. But until Shinya published, we didn't have KLF4. We had all the other factors, but not KLF4. We didn't know about that. And so once he published, then we knew the last factors, to say. And then it took only a few weeks, because as you know, reprogramming is actually doesn't take that long. And then we had, our own iPS cells as well in our hands. So that, those were really, really moving moments.
Yeah, it drives home. I mean, for you, Arun, who you've lived your whole life as a scientist, it seems, taking iPS cells for granted. I think that really drives home that answer of how unexpected, how crazy it was, how, great an advance it was, and why it won the Nobel Prize for sure.
Yeah, and the fact that it was reproducible, like what you said, it was something where that paper was published, a lot of people didn't believe it, but then you brought it into your own lab and you're like, whoa, this actually works, right? That's part of the reason why it was so exciting.
I will never forget this. I think it was there was a series. I mean, I was a postdoc, so I didn't attend a lot of meetings, right? So, but Rudolf, my mentor, went to all the meetings. I think it was in ICCR meetings, some Gordon conference, maybe, I don't know, where Shinya every few months or so, every few weeks, disclosed one more factor. The first meeting said, "Oh yeah, OCT4 is one of the reprogramming factors." Well, sure, OCT4, thanks, and the others, and the next, oh yeah, SOX2 is another one. Oh sure, I mean, thank you. So he didn't, so I think at some point he disclosed CIMIC, but he never disclosed KLF4 until it published in Cell.
Yeah, it was, I think, a defining moment for a generation of stem cell biologists. I mean, honestly, that's like IPS is the reason why I decided to go to grad school and ultimately go to Stanford for my PhD. But like that's why I got into the field, and I think there's so many other folks like that. Folks in my generation didn't know a time before IPS. That kind of tells you something, right? So I guess talking a little bit more about the direct programming side of things, of course, another foundational study in the field, really a milestone study coming from your lab. This is a paper, of course, that was published in Nature back in 2010, led by Tom Verbuchen, who I believe was a grad student at Stanford at the time in your lab.
Yes, he was a rotation student.
Wow.
I know you know this. He found the cells during his rotation.
Wow. That's a great rotation.
whenever a new rotation student comes in, you can be lucky and get a nature paper out of your rotation, but it's rare.
That would have been amazing if he was like, that was a good rotation, but I'm thinking of going somewhere else.
Wow, that's an amazing story. I had no idea. Yeah, but it was such a foundational finding, and I don't know if you actually remember, we had a really small paper that we published together on direct reprogramming of cardiomyocytes to neurons, partial reprogramming. It was more exploratory than anything else, but that was kind of cool. I think it was with Sean Wu's lab at Stanford. But I guess tell us more where we are right now in the realm of direct reprogramming, especially when it comes to making neurons. I mean, we've talked about it a lot on this show as a complement or maybe an alternative to iPS technology in terms of mass producing cells like cardiomyocytes and neurons that otherwise might be hard to make. So I guess tell us more about the current state of the art in neuronal direct reprogramming. And if you want, maybe a little bit more about the translational applications as well.
Yeah, so I think one big impact it actually had on the iPS cell field, which was not quite expected when we started this, because it turns out, and this is not a new concept, of course, to use transcription factors to program iPS cells, but I think our work really has sort of made it so clear and sort of so obvious for everybody. And in our lab, we spent a lot of effort now also optimizing transcription factor combinations just to differentiate. I guess we should say forward program perhaps iPS cells to very specific lineages, and that really has taken off. And these transcription factor protocols, in particular the one transcription factor NGN2, that magically almost converts iPSs into neurons sort of overnight, has really been widely accepted and used in both in industry and in many labs. It's very rewarding for me to sometimes to go through poster sessions where a lot of posters use these NGN2 cells. Oh wow, this we really helped a lot of other labs getting more reproducible and large numbers of neurons from human iPS cells. So that was a little bit of an unexpected outcome. And when it comes to the true, direct reprogramming of somatic cells to neurons, it's a hard thing to do. A fibroblast doesn't want to be a neuron, and I guess that's why it was also such a big finding, right, in the beginning, that it is possible at all. And it turned out that after an initial hype, there were a lot of labs like jumping on this and contributing to some proof of concept, findings. You can, for example, you can make different cell types. You can also get it to work with human cells and so forth. But once this wave of proof of concept papers were over, then it became to optimize the reprogramming to actually be useful, right? And then many labs left the field, and we were again kind of alone. And as a single lab, it's hard to really drive, technology. But there were, we were not completely alone. So I think I do have the impression that there's though more and more people coming back to this and sort of joining our efforts to optimize and make things more efficient and get more mature cells through the direct reprogramming process. Because it turns out that there might be some important advantages to use a fibroblast or other somatic cell directly reprogrammed neuron as opposed to iPS cell-derived cells. As we have learned over the years, is when you reprogram cells to iPS cells, you really rejuvenate the cells, which is a totally fascinating, separate, I think, piece of biology. And big efforts like the ITOS lab efforts, for example, I'm sure you've heard of, are really based on this finding. But the disadvantage of that is that if you want to study aging type diseases, and that's one of the main reasons why we want to have neurons from patients, is to actually study things like Alzheimer's disease and Parkinson's disease. And those happen in many decades old neurons. And turns out these iPS-derived cells, they're really very young and very healthy and sturdy. Their proteasome activity is like through the roof. Their protein turnover is like this. And that is a very different situation than what you have in an old neuron, right? And it turns out that this is really what Rusty Gage pioneered, is that when you reprogram fibroblasts directly to neurons, you have a much more similar sort of cell biological state sort of an aged cell biological state when you reprogram neurons directly. So I think mostly for that reason, there's really a renaissance, I would say, of using the direct reprogram cells from somatic cells, and sort of a renewed urgent need to make the reprogram system also more efficient. For iPS cells, there was such a huge and obvious need and so much effort from really the whole field that the technology has just been exploded, and it's every high school student doing a summer rotation can reprogram iPS cells with I don't know, crazy efficiency, right, in 10 days. So that effort has not been devoted by the field to direct reprogramming. So that's why it's still hard to make the cells, and they're relatively inefficient. And you need to have a lot of patience to actually have enough material to do proper experiments and so forth. So it's if you do it, you really have to want to do it, and then it's a commitment.
Well, no one better than yourself in your lab to sustain that effort and lead that renaissance. And I like what you said there, because that's the way it is with so many things in science, right? The trends, or you get a big idea and everyone's like, oh yeah, that's a great idea. Let me try that in my system. And there's all this push, but often it's a new idea. It's not really ready to go into full, translational application. It's like, I don't know, I'm aging myself here, but Apple way back in the day came out with a Newton. It was a handheld PDA, personal, whatever the D stands for, DA, I don't know. Arun knows, I'm sure. And nobody bought it. It didn't work because it was too cumbersome and whatever. And then the iPhone came out, right? So like the tech caught up, and then you get the world-changing device. It's widely adopted. And I feel like the direct reprogramming now, as you described, is in the midst of that renaissance, because the technology to support, as you said, probably to support iPS has matured. And now maybe some of those tools can be deployed in the direct reprogramming system. There's a strong rationale for doing it, as you described there too. But in terms of the trends, I say, there's the seminal investigator there. It makes sense that you'd stay in that field, but also you've done some other things. You've really diversified the translational scope of the lab. We just talked to Jonathan Thomas, who's a new CEO of CERM, on the show. So I got to look into some of the CERM awardees, and yourself among them, for multiple awards. But the one that caught my eye in particular was this project, I think you just published about this developing GMP compatible therapy for epidermolysis bullosa, which, I mean, you could draw a line between that and neural and direct reprogramming, I guess, but that's pretty far afield. So I'm impressed that you've got such a broad and wide scope. But tell us a bit about that, and also related, since we're talking about CERM, how does a unique format of CERM and/or California-based philanthropic funding, how does that enable this work specifically?
Yeah. Thanks for the question. It's a great topic to talk about. So so this project is one of the first projects that I started at a lab. It actually was, I think, the very first talk I gave it was a Cancer Bio PhD program retreat. I think I didn't have the keys to my lab yet, so I came straight from my postdoc from Boston to this retreat. And of course, I was the new kid on the block, so I was allowed to give a talk during the retreat. And so this was, now I'm dating myself, but this. So this was pretty much right after, the iPSs were discovered, like a year after the human iPSs were published, And I just, put out a couple schematics. This is what we could do. Now we can reprogram. We can do genetic engineering. We can make all type of cell types in principle. So that would be fantastic to develop a really powerful cell therapy. We don't have to worry about random mutagenesis with retroviral gene transfers, right? It has been really a huge blowback in gene therapy a decade before. So we can go in, the CRISPR wasn't around yet, but still we envisioned, you can do any type of genetic modification, including just changing a single base pair, which is the cause of so many horrible diseases. So I have this slide up there, and then I move on to showing the basic science. And there were a ton of people in the audience, including Tony Oro from dermatology department, and he's immediately clicked like, I have to talk to this guy. So and then right after my talk in the break was in Asilomar. I don't know, you know, this beautiful sort of conference center very close to the ocean. There's a beautiful beach. So it was beautiful sunny California weather. You know, it's sometimes in November, some wintry month otherwise. And so we walked on the beach and just ironed things out. Like, hey, we have this disease. It's called epidermolysis bullosa. It's a horrible skin disease. It's a blistering disease. It's caused by mutations in collagen 7, which is essentially the glue between the epidermis and the dermis, so between the upper layer and the lower layer of the skin. And when this gene is missing, or this protein is missing, the upper layer of the skin just comes off in blisters. And these poor patients are born that way and develop wounds wherever you touch them, right? And these wounds cannot heal because the skin can attach. And eventually they, with highly proliferative and inflammatory environments in these wounds, eventually gets a horrible, aggressively malignant squamous cell carcinomas and that die from this before they reach 20 years. So it's a horrible disease. And the dermatology department has been working on this disease since decades, even already then, and had already started a cell therapy or a project based on retroviral ex vivo type gene delivery, which, by the way, has come a long way. And they're about, I think they've just finished the phase 3 trial, which also looked very good. But from the very early on, it was already clear the limitations of trying to expand a somatic cell, right? Because it affects the entire skin, the entire surface of a person's body. So scaling is a big problem. And of course, the retroviral gene transfer is clearly not ideal, right? And both of these limitations can be solved with the iPSCL technology. So from this very moment on, we collaborated with Tony Oro's group. And as you mentioned, it's still going on. And we just published Nature Communication papers a few months ago, and had various flavors and versions of this project. But in the end, it's the same idea, using iPS cells to gene correct the underlying mutation in an autologous way. So we are facing, of course, the harsh conditions in the sort of biotech finance market, which they don't like autologous approaches. Unfortunately, they really like off-the-shelf products. And in the end of the day, we need to treat patients. So we need a commercially viable product. So even though we have the highest aspirations, we know the technology is there, it's possible to do. But if it's not practical for a for-profit entity, which is the only way to get drugs developed, unfortunately, in the end of the day. So we will see. Maybe we have to make some compromises, but we don't want to. And so at this point, so this is the second sort of area that you mentioned. We are very fortunate to have STERM around here. It's been really, really an enabler of exactly this type of problems, that you can really incubate potentially transformative technology and idea very long in an academic lab and bring it very, very far, almost maybe even into a phase 1 clinical trial, which will then hopefully be sufficient for investors that have to make money, right? Venture capitalists are essentially bankers, right? They're investment bankers. They it's just a different modality how they try to make money, but that's their job. Their job is not to make drugs or cure people. Their job is to make money. Of course, they try to do this while helping people. That's, I think, their motivation. But in the end of the day, they get out of business if they don't have a good return on their investment. So even if they wanted, right, they cannot really invest in very early stage and very risky new therapies. So it's really up to us in academia to try to drive this as much as possible. But as you know, it's very hard to get funding for something that's not so sexy. The idea is sexy, and the first proof of concept, is published relatively quickly. But then actually develop this in a way that the FDA would be happy with this and as efficient as possible, because you have to think of the cost of goods. It is very high. It doesn't probably help if you develop a $10 million therapy for one patient, right? So that's not commercially viable. So you have to already think in optimization processes and how to streamline things. And that is sort of boring, mundane work, right? Grad students and postdocs are not really so interested in that. Some are actually, but most of them are really more excited by like the breakthrough discovery, but not like optimizing a differentiation process or a programming process or gene editing process. So that's why CERN was really, really great to have around here, because that is exactly sort of this niche type or this little bit of a vacuum actually of area that needs funding if you want to develop these type of therapies. Really long breath.
Absolutely. I think, we're all very grateful for CERM. And in addition to funding the science, they also fund the education, of course. It's thanks to CERM that folks like myself and many others have been trained as stem cell biologists here in the state of California. So we're obviously very grateful to CERM. And another area that they're very interested in funding is, neurodegenerative diseases like Alzheimer's, for example. I used to work in Clive Svensson's lab. He has got a huge focus on this particular area as well. You actually had a stem cell paper recently exploring a unique cell therapy approach to potentially restore microglial function in a mouse model of Alzheimer's disease. I mean, you said it yourself in that paper that, quote, Alzheimer's disease remains one of the grand challenges facing human society, which is definitely true as society gets older. And
Oh, I forgot I wrote this in the paper, but it's true. That's what I always say.
There you go.
Yeah.
So tell us more about this work, how you use the systemic hematopoietic cell transplantation to actually restore microglial function in this mouse model.
Yeah. So starting as a medical student, I was fascinated by stem cell biology, and that's one of the reasons probably why I was seeking out labs that would be working with pluripotent stem cells really, really early on. But really with the ultimate idea in mind of regenerative medicine, and that we can do something for the CNS and transplant various types of cells into the brain. Both early on, I had these both directions in mind, both for actual regenerative medicine in the proper sense. There's a lost neuron or a neuron that's degenerated, and let's just try to replace that by transplantation, very much along the lines of what is being done in Parkinson's disease now, right? Where it's a very defined dopamine neuron population that is degenerating, and people attempt to literally just replace that one. But the other aspect of cell therapy is not necessarily to replace something that is lost and regenerate that same system again, but actually to transplant cells into the brain that may be somewhat artificial and not physiological components of the brain, but still be therapeutic in the broadest sense. So made it both ways would make sense, right? And I think have a perfectly justifiable reason to put cells into the brain. So along my career, starting as a medical student, I remember I thought of all kind of and tested all kind of neural cells, of course, various types of neural stem cells, neuron stem cells, oligodendrocyte precursor cells, some other glial, some maybe by lineage potent glial progenitor cells, and so forth. And this more or less, even the OPCs, the oligodendrocyte precursor cells, which are much more migratory than other cell populations. But overall, it was quite disappointing to see that when you put these cells in the brain, they by and large stay where you put them. So in most cases, you don't really get a proper incorporation of these cells. What you rather get is that these cells usually proliferate a bit more and then generate a really like a circumscribed tissue on its own that is integrated, it's sitting in the brain. But it's really, the brain is more like an incubator for this exogenous tissue, right? So it's not really they will connect, right? So the neurons in the graft, they will grow axons out, and both ways, the host neurons will connect synaptically, make synaptic connections. We and many other labs have shown that. But still, it's not what I sort of had envisioned, and certainly not a good distribution in the brain. So at some point, I had a new postdoc who had been working with microglia before, more on the sort of disease modeling side of things for neurodegeneration. As you know, the human GWAS data are very much pointing to microglia. So the Alzheimer's field has been sort of woke up and has to try to understand what the role of microglia in neurodegeneration is. So that's been a big focus since several years. But we really had this sort of eye-opener. Hey, microglia are also important cell type in the brain. We never really thought about them, but they're not neuroectoderm derived, not the most elegant, I guess, neural cell type. But still, they're a super important component of the brain, and they have a completely different origin. Obviously, they're hematopoietic cells. And we got reminded that many people have already done bone marrow transplantation before and tried to map what the cell contribution into the brain is. And we were not exactly clear by just reading all this literature, how efficient is this really, this incorporation of myeloid or microglia-like cells derived from a bone marrow transplantation graft into the brain. So that's when we really started. Let's just map this out in our own hands, in our own lab, and we essentially ended up optimizing this bone marrow transplantation procedure in a way that we got a reliable, close to 100%, chimerism every time we transplanted a mouse, and that was really the first time I saw the section that my postdoc showed with the microscope, full of these transplant cells in the entire brain, homogeneously distributed, just like the microglia are, right? I was really blown away. So it's not what I expected, beautifully fancy, complicated neurons, for example. It's a microglia, but still a great way to use these cells as vehicles to deliver biologics, for example, into the brain, or try interfering with local, maybe disease processes. So we thought, we really should explore this more, and that I'm happy that a bunch of new trainees that joined the lab since have shared this excitement with me, and we have a significant group in the lab that is really focusing on this aspect now, also including human iPS-derived microglial precursor cells, getting this more efficient, and things are looking really, really promising. So there's still hurdles. The bone marrow transplantation is kind of toxic. I mean, if you want to make it efficient, it's actually quite toxic. So it's clear we have to get rid of the irradiation or the chemotherapy conditioning that is required to condition the bone marrow for that to work. So that's a huge issue for us to try to work this out. And we think we have some ideas, and the things look kind of promising. I want to stay optimistic, because we actually don't want to replace necessarily the bone marrow, right? All we want is replacing the microglia in the brain, so we don't necessarily need to condition the bone marrow that way. So we have some ideas, and hopefully we'll be able to develop a similarly efficient microglia replacement transplantation approach without this toxic preconditioning.
Yes, I mean, hearing you describe that, I got a little chill, because that's what it's all about, right? There's everything there. You have a great idea and like a concept where you're like, oh, wow, but you got to test it. And oftentimes you test it and it gives you a result you weren't expecting. It's still awesome, right? And you get that moment, which we all seek as a scientist, that like aha or whatever, that seeing something for the first time. But I wonder, I mean, you've made it as a scientist. You did it. You got it all. You got a nice lab, as you're describing. I had some good funding. You got your work actually going into humans and having potential, like all the stuff that I think scientists dream of, including like the seminal contribution that puts you on a short list. But looking at your portfolio of research, and as you describe it, the scope is so much greater. It's expanded, right? And that's what you expect. You make your own splash, and then you extend it by assembling a exceptional group of trainees and collaborators. But I'm curious because I hear you describe it, and I see that in you. I can see you still get that jolt. You still have that feeling when you see the result, but you're seeing it for the second time now. The postdoc sees it, and they bring it to you. So I got to ask, do you ever get back in a TC or otherwise anything to hands-on experiment, get the pipettes, get your own eyes in the scope? I mean, I have a tiny lab, a much more modest profile, so I kind of have to get in the lab to get productivity up. But I love it. Every single day, I love going to the lab and looking at my culture, seeing what's going on, incremental differences, maybe some unexpected results. I love it every day. So you got a lot of responsibilities, commitments. Doesn't leave a lot of time for the wet work. But if that is the case and you don't get wet, do you miss it? And if not, is it because there's this equivalent that you get as a mega PI that is probably the dream, where you get to scale up that jolt? You get like a jolt every day with all these postdocs coming to you with results.
I wish. No, the jolts are few and far in between. Even as a PI running at a sort of 15 people lab, which is what I do. Yeah, there are so many questions. I don't know where to start. So first of all, I guess the question: do I get back to the lab? Unfortunately, not much. It's as I said, I have 15 people in the lab, and they all have more than one project. And I learned I can help them much more by not doing my own projects. And just be there for them. Of course, grant writing is a huge task. Especially after the pandemic, I feel everything has become so much more expensive. Salaries got up, which is good for the lab people, but I have to raise the money to pay all of them. So that's of course a big job, and just requires just a lot of time. So unfortunately, I'm not really in the lab anymore at all. But I still can do it. It's in the beginning, when my lab was small. Of course, I was still pretty much a senior postdoc, training rotation students and so forth, and showing people how to run a PCR. But relatively quickly, though, because we were lucky and we published, we had this finding very early on, the director of programming. So there was a prominent Nature paper, and I got a lot of funding right after that, which is, by the way, I also learned an interesting lesson there, because I wrote the same amount of grants, but all of a sudden all the grants got funded, or none got funded. Anyway. So so I had actually early on a lot of funding, so I had to hire people. So that's why I grew actually a lab quite quickly, pretty much to the size that I have now ever since. And that really got me out of the lab very quickly. But at some point, a couple of years after this, as I don't know why, but I sort of had to, I wanted to, or was some reason why I actually had to go back to the tissue culture. And I'm super nervous. Now all my grad students and postdoc were like watching me. Oh no, the PI is going to the lab. Like, what's going to happen? And I thought, oh, I'm sure I will screw everything up. I don't know where the pipettes are. But turns out this is hardwired. I think I probably spent, I don't know, millions of hours during my postdoc and before, in tissue culture. So this is subconscious. So it was no problem. So I'm still good at it. And as I said, it's totally enjoyable, but it's just not time efficient.
Yeah, that's fair. And I'm kind of running into this same thing as somebody who's started a lab recently, and it's a small team of like 5 or 6 people. Sometimes it's just better to get out of their way, especially if you haven't been doing it for a while. But you're right. I mean, like I jumped in the hood a while back, recently in the last month, and it's almost like muscle memory. If you've done it so many times, it's sort of hardwired into your muscles to be able to pipette and change media and that sort of thing, right? So thanks so much, Marius, for being here. It's been awesome catching up with you. You're such a foundational member of our field, such a pioneer in our field, and somebody we all look up to. And before we let you go, we have a couple peripheral questions for our listeners to get to know you a little bit better. I think these are sort of lumped together because you may have a similar answer for both or a single answer for both. So basically, if there's one hobby you wanted to pursue but were never able to, what was that hobby? And I guess similarly, if you were not a scientist, what would you be?
Yes, yes. And I have a hobby like that was always competing with my decision to become, a biomedical scientist. Or initially, I studied medicine, so it was even on the possibility to become a physician. So that would be music. I, for some reason, I don't know exactly why, but I was very early on drawn to music. As an, I guess, early teenager or so, I played a bunch of instruments and started composing. And I was pretty good at it, actually. In high school, I got together with a couple other folks, actually, and we composed an opera even that was performed. I was at the time in Bonn, Germany, and Cologne is a pretty big city, and they have an opera house, and they actually performed that opera. So it was a really cool moment as well to hear something that came out of your own mind, and everybody in the room hears the same music. That's really, really cool. And I was really debating to study composition, but I also had, of course, my huge passion and interest in biology or the natural life sciences, really, and made that decision. So I wish I had more time to spend on, in particular, writing music. I still play, and that's fun. I change instruments to make it a little easier. I don't have so much time to practice. So I still can play music, but writing the composition part is just huge time commitment. You can imagine, for example, if you write a piece for an ensemble or so, every single instrument needs their own, notes. Just writing that all up is really a huge time commitment. If you're not, I guess I'm not as gifted as the famous composers we all know, right? So they probably, somebody like Mozart probably composed in dreams. So I'm clearly not like that, but I really, really enjoyed it with a huge time commitment. So that's the one thing it's a little sad that I had to sort of give this up. It's just not possible to do both.
Well, I have to say it's a new one for me. And like most things in your body of work, it's high level, operatic composition. Wow, intense. But I see it. And I love this question because it gives an insight into that, sliding doors, alternate path. And I see the symmetry there as you describe it, of like conceiving of something and not even knowing if it's good. Until it is brought to life in the world, and then in that moment actually having I guess it's a similar jolt. So you've got a good hobby there, and it's not too late. Maybe you'll make some time when you retire from innovation and get back into the arts, but not too soon. We still have need of you, Marius, so get back in the lab. Well, not actually back in the lab, but keep your efforts going. And thank you again so much for sharing an update. It's been like over a decade since you've been on the show. I wasn't on the show then. So this is a first for me, and this is a real great pleasure I think I can speak for Arun and our listeners as well. Thanks again.
Thank you. It was a pleasure.
All right, you guys, that brings us to the end of a pretty spectacular episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including a summary and links to all of the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. Here we are in the new year. New episodes abound. Stay tuned, you guys. We got another one coming to you in a couple of weeks. Until then, thank you so much for listening.